So far, we've talked about the secondary lymphoid organs, in which B and T cells meet to do their activation thing. But I haven't said much about how these cells know to go there. Immunologists call this process lymphocyte trafficking. So here we will be discussing lymphocyte trafficking in detail. It's a very important concept in immunology. I will be explaining this by using interesting analogies. So you have to follow me to make your concepts crystal clear. In a human, about 500 billion lymphocytes circulate each day through the various secondary lymphoid organs. But these cells don't just wander around. No, they follow well-defined traffic patterns, which maximize their chances of encountering an invader. Now, an important thing worth mentioning here is that the traffic patterns of virgin and experienced lymphocytes are different. So, let's first look at the travels of a virgin T cell or naive T cells. Virgin or naive T cells, as the name suggests, are those T cells that have not been primed, meaning they have not encountered their antigen yet. So T cells begin life in the bone marrow, and then we can say they are educated in the thymus. When they emerge from the thymus, virgin T cells express a mixture of cellular adhesion molecules on their surface. These function as passports, you can say, for travel to any of the secondary lymphoid organs. For example, virgin T cells have a molecule called L-selectin on their surface that can bind to its adhesion partner, glycan-1, which is found on the high endothelial venimals of lymph nodes. This is their lymph node passport. Virgin T cells also express an integrin molecule, alpha-4-beta-7, whose adhesion partner, MAdCAN-1, is found on the high endothelial venimals of Peyer's patches, and the lymph nodes that drain the tissues around the intestines, the mesenteric lymph nodes. So this integrin is their passport to the gut region. Equipped with an array of different adhesion molecules, inexperienced T cells circulate through all of the secondary lymphoid organs. This makes sense, right? Because the genes for a T cell's receptors are assembled by randomly selecting gene segments. So there is no telling where in the body a given naive T cell will encounter its cognate antigen. Well, in the secondary lymphoid organs, virgin T cells pass through fields of antigen-presenting cells in the T cell areas. There, these T cells seem like checking the billboards on several hundred dendritic cells. If they do not see their cognate antigens advertised, they re-enter the blood, either via the lymph or directly, in the case of the spleen, and they continue to recirculate. On average, naive T cells make this loop about once a day, spending only about 30 minutes in the blood on each circuit. A naive T cell can continue doing this circulation thing for quite some time, but after about 6 weeks, if the T cell has not encountered its cognate antigen, presented by an MHC molecule, alas, it will die by apoptosis, lonely and unsatisfied. In contrast, those lucky T cells that do find their antigen are activated in the secondary lymphoid organs. These are now experienced T cells. Also, these experienced T cells carry passports, but they are restricted passports. Well, this is because, during activation, expression of certain adhesion molecules on the T cell surface is increased, whereas expression of others is decreased. This modulation of cellular adhesion molecule expression is not random. There's still a plan here. In fact, the cellular adhesion molecules that activated T cells express depend on where these T cells were activated. In this way, T cells are imprinted with a memory of where they came from. For example, Dendritic cells in Peyer's patches produce retinoic acid, which induces T cells activated, there to express high levels of alpha 4 beta 7, the gut-specific integrin. As a result, T cells activated in Peyer's patches tend to return to Peyer's patches. Likewise, T cells activated in lymph nodes that drain the skin upregulate expression of receptors that encourage them to return to skin-draining lymph nodes. Thus, when activated T cells recirculate, they usually exit the blood and re-enter the same type of secondary lymphoid organ in which they originally encountered antigen. This restricted traffic pattern is quite logical. After all, there is no use having experienced helper T cells recirculate to the lymph node behind your knee if your intestines have been invaded. Certainly not. Because you want those experienced helper T cells to get right back to the tissues that underlie your intestines to be re-stimulated and provide help. So, equipping activated T cells with restricted passports ensures that these cells will go back to where they are most likely to re-encounter their cognate antigens, be it in a Peyer's patch, a lymph node, or a tonsil. Now, of course, you don't want T cells to just go round and round. You also want them to exit the blood at sites of infection. That way, cytotoxic T lymphocytes can kill pathogen-infected cells. And helper T cells can provide cytokines that amplify the immune response and recruit even more warriors from the blood. To make this happen, experienced T cells also carry combat passports, the adhesion molecules, which direct them to exit the blood at places where invaders have started an infection. These T cells employ the same roll, sniff, stop, and exit technique that neutrophils use to leave the blood and enter inflamed tissues. For instance, T cells that gained their experience in the mucosa express an integrin molecule, alpha E beta 7, which has, as its adhesion partner, an addressin molecule that is expressed on inflamed mucosal blood vessels. As a result, T cells that have the right training to deal with mucosal invaders will seek out mucosal tissues which have been infected. In these tissues, chemokines given off by the soldiers at the front help direct T cells to the battle by binding to the chemokine receptors that were expressed on the surface of the T cells during activation. And when T cells recognize their cognate antigen out in the tissues, they receive stop signals which tell them to cease migrating and start defending. In summary, naive T cells have passports that allow them to visit all the secondary lymphoid organs, but not sites of inflammation. As a consequence, the entire collection of virgin T cells travels through the secondary lymphoid organs and greatly increases the probability that these T cells will be activated. The reason that virgin T cells don't carry passports to battle sites is that they couldn't do anything there anyway. They must be activated first. In contrast to virgin T cells, experienced T cells have restricted passports that encourage them to return to the same type of secondary lymphoid organ as the one in which they gained their experience. By recirculating preferentially to these organs, T cells are more likely to be re-stimulated, or to find cytotoxic T lymphocytes and B cells that have encountered the same invader and need their help. And of course, experienced T cells also have passports that allow them to exit the blood at sites of infection, enabling cytotoxic T lymphocytes to kill infected cells, and TH cells to provide appropriate cytokines to direct the battle. This marvelous postal system, made up of cellular adhesion molecules and chemokines, ensures delivery of the right weapons to the sites where they are needed. B-cell trafficking is roughly similar to T-cell trafficking. Like virgin T cells, virgin B cells also have passports that admit them to the complete range of secondary lymphoid organs. However, experienced B cells are not as migratory as experienced T cells. Most just settle down in secondary lymphoid organs or in the bone marrow, produce antibodies, and let these antibodies do the traveling. So here we end our discussion about lymphocyte trafficking. Just to conclude the whole video, and to give you a reminder again, the secondary lymphoid organs are strategically placed to intercept invaders which penetrate our barrier defenses. During an infection, rare T cells must find antigen-presenting cells, which display their cognate antigen, and also B cells must encounter the small number of helper T cells that can assist them in producing antibodies. Now, the secondary lymphoid organs make it possible for antigen-presenting cells, T cells, and B cells to meet under conditions that favor activation. Well, the trafficking of immune system cells throughout our body is controlled by the modulated expression of adhesion molecules on the surface of these cells. And virgin and experienced lymphocytes move in different traffic patterns that we have just discussed in detail. Goodbye for now. But you can find more such detailed videos only on sqadia.com.